The other thing you can do is use a spectrophotometer. Okay, so you can take this and you can put your sample into a spec and read the reading. In general, for at least E. coli, an absorbance unit or an OD of 1.0 is about 10 to the 9th cells per mil. Okay, that's a ballpark figure. Okay, so you need to have a little cuvette that fits this little machine. You need to have a blank. So your blank is going to be whatever the cells are growing in. In this case, they're growing in some TSA, and I've got some TSA liquid here. So I'm going to fill this thing up. It doesn't even take a full mil. 500 microliters is sufficient. But you use sterile technique. You know, this is a plastic bottle, so I can't flame it. And you note that you don't want to touch the outside of this. You note there's usually a little arrow there on the top, and that designates where this thing goes. You want this thing to go down, in this case, vertically like so. And in this case, with this machine, you hit blank. And there's our blank. Okay. Now, you take your sample. New tip, sterile technique. Put it in and hit read. And we get 1.17 in this case, okay? So when we plate these out or we look at our data, we'll see if this number actually correlated. That means to be a little over 1 times 10 to the 9th cells per mil if this is correct. You're going to use this when you do your growth curve in a little bit. And that's going to rely on you using both the spectrophotometer and taking a sample and finding what that number is, recording it, and also taking your that same sample as it's incubating and doing serial dilutions at each time point. Now, how do you use this in a court of law? You're getting what are called colony forming units. And what that means is the number of colonies you see on a plate. So what you've done essentially is you've taken a sample, you've taken and made serial dilutions out of this. So you've done a 1 to 10, a 1 to 100, a 1 to 1,000, a 1 to 10,000, okay, etc. You've then plated from those 100 microliters onto plates, okay? And so you have a plate here, 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 okay? So this guy is the 1 to 10. This guy is the 1 to 100. This guy's the 1 to 1,000. This guy's the 1 to 10,000. This guy's the 1 to whatever comes after that, etc. Okay. Now, you will, after 24 hours or 48 hours, you will count the number of bacteria you see on these plates, the bacterial colonies, the colony-forming units, CFUs. Okay. And what you're going to find is, at the low dilutions, you're just going to see a lawn of cells. It's going to be a lot of bacterial colony there, okay? And that's not going to help you much, okay? Maybe at this one, you're still going to see a bunch of colony that you're not going to be able to look at individuals. Over time, though, you're going to start to see colonies showing up and individuals, and you're going to start to be able to count those. But what you're looking for, your target, is a plate that has from between 30 and 300 CFUs on it, okay? So let's pretend, in this case, we've got and we count these colonies, we get a value of 62. Okay. When we look at these colonies on this last plate, we get 1, 2, 3, 4, 5, 6, 7, 8, 9 or so. We'll just say 9 CFUs. Okay. What does that now? How do we use these numbers? We can't use any numbers here. We don't have any. In fact, those are called. Well, that's a lawn. I really wish markers would work. That's a lawn. This is a lawn. This one is not quite a lawn, but it's still too many to count. So you say TMTC for too many to count. This one is spot on. That's something that's countable. It's between 30 and 300. This one is below 30, so it's called TFTC for too few to count. Okay? So how do you use these numbers then to calculate how many cells per mil you got? Well, take the one that you can count, in this case 62, and you know it was in the 10 to the minus 4th dilution. Here's 10 minus 3rd, so it's a 1 to 10,000 dilution, okay? So you come up here and you do a little formula, which is super easy. It is the CFU, okay, times the dilution, okay, whatever the dilution is. In this case, well, we'll just say times the dilution factor. All divided by how many mils you plated. And in our case, standard, we're putting in 0.1 mil, which is the same as 100 microliters. That's what we put down on our little plate. That's going to give you CFU per mil, or cells per mil. Because remember, a CFU, we're saying a colony is the same as, started out as one cell. So all you do is multiply 62 times 10 to the 4th divided by 0.1. And that would give you your cells per mil. Okay, everybody, so this is 24 hours after I did our platings and everything we did yesterday. And here are some of the plates laid out for the serial dilution. And you can see what I meant by when I was drawing on the board. This is, our 1 to the 10th dilution. It's way too many to count. It's basically a lawn of bacteria, all these colonies, and you really can't tell one from the other. You certainly can't count all these. Here's a 1 to 100, a little better, but still not countable by any means. 1 to 1,000, getting better, okay? I still wouldn't want to count these. And if you decided, like if there was a situation where you didn't have anything else that was countable and this was all you had, what you could do, which is kind of illegal, but why not? In this case, you could sort of split this sucker into quadrants, say split it into 4. Approximately equal, and you could say count the colonies that are in one quadrant and multiply by 4. Okay, that's really not legal, but it's it would give you at least an estimation. Then you'd put in your lab book or your report that this is a gross estimate, and here's why. Okay, but still. And then we get down to number 1 to 10,000, and you can see what I've done is I've taken a Sharpie and I just held this up and just tapped on the back and counted in my little head all these little colonies. And there's different colony types. It was a mixed culture because you got these big puffy ones here and you got these little tiny ones that you can see under the black dots. And I got 235 at a 1 to 10,000 dilution. Okay, so let's see. That means I got 235 CFUs. I multiply it by my dilution, which is 10,000, and I divide by 0.1. and I put it into notation so that we scientists can understand it. And I get 2.35 times 10 to the 7th cells per mil. Okay? Fantastic. I believe it. This is looking at live cells, and every single colony is an individual cell. That's wonderful. Now, when we did our absorbance reading on the spec over here, we got like 1.17 or something like that. I can't remember. And we said that one absorbance unit was 10 to the 9th. So let's see. 1 times 10 to the 9th times I guess 1.17. I think that was right. Okay, so, and divided by, of course, 1. Just setting up a proportion. So the estimate from the spec is 2 orders of magnitude higher. Okay, one was 10 to the 7th, one was 10 to the 9th. Okay, so I want you to think for a little bit why that might be. Okay, obviously, you know, this one is actually more realistic because this is what you see, this is what you count. These are actual viable cells. So I want you to think about why you'd get a number from the spec reading using a little spectrophotometer that's grossly overestimated. Okay, so think about that a little bit. But overall, this shows you, you know, the basics of how this works, and it worked really, really well.